Hello everyone, and welcome to the National Association of Plant Breeders webinar series, The Science of Selections. My name is Anna Levina, and I'll be serving as your host for the webinar. Today's presentation will be given by Dr. Istvan Rajcan from the Department of Plant Agriculture at the University of Guelph in Canada. His research focuses on seed compositional traits, for example, modified fatty acids, isoflavonoids, and vitamin E, for food and industrial use, and exploration of genetic variation for yield and disease resistance traits using exotic germplasm as well as organic soybean breeding. Today, Dr. Rajcan will be speaking about the soybean breeding and genetics in his presentation entitled Soybeans More Than Yellow Seed. After the presentation, you will have the chance to ask questions. We'll answer as many questions as we have time for the question period. And now I'm going to switch control of the screen to our speaker, Dr. Rajcan.
Thank you very much, Anna. And I'd like to start by thanking the National Association of Plant Breeders for kindly inviting me to participate in this seminar series. And also in particular to Dr. Aaron Carter from Washington State University who invited me to take part in this series. In my talk, I will give an overview of my research program, of how we do soybean breeding in my program, which is just one of many ways that it can be done. I'll give some examples of the traits that we have been working on. I will also follow the script that I've been asked to follow in terms of explaining the basics of soybean breeding to a wider audience, and give some examples of some graduate student projects that are ongoing in my lab. So before I go into the meat of the talk, I'm just going to introduce myself a little bit. So I did my bachelor's degree at the University of Novi Sad, which is in Serbia, and at the time it was still called Yugoslavia. My bachelor's thesis topic was in sugar beet physiology, so I almost became a plant physiologist, but then an opportunity arose after I graduated with a bachelor's to go into sunflower breeding, and that's what I did for the following several years at the University of Novi Sad Institute of Field of Vegetable Crops. In 1991, I moved to Canada and started a master's degree, which I did for about a year, and then transferred directly to a PhD program, which I completed in 1996. Since I came to Ontario, to Guelph, Ontario, there was very little sunflower being grown here, so I switched my interest to canola breeding and completed my dissertation in the genetics of modified fatty acid composition in canola or Brassica napus. After graduating from Guelph in 96, I moved to Saskatoon, Saskatchewan in Western Canada and became a specialty oil canola breeder with the company Viterra. In 1998, an opportunity arose for me to come back to the University of Guelph, and I accepted a position at the time as an assistant professor in soybean breeding and genetics in 1998, and I've been here having fun ever since. I just also wanted to say a couple of words about the University of Guelph, as I don't know who exactly the audience is. I assume, or I understand, that I may be the first Canadian plant breeder talking about or participating in the webinar series. So some of you may have heard of the University of Guelph, others may not. So I'm just going to say a couple of words briefly about our university. Our university actually started off as Ontario Agriculture College, which was established in 1874, and it is the oldest agriculture school in Canada. For a long time, it offered degrees in agriculture, first as a one-year program, then 2 years, and later a full degree. And we added a master's degree in the 1920s, and a PhD degree came later. In 1964, the university was actually established as a separate University of Guelph, of which OAC, Ontario Agriculture College, was one of the founding colleges. So since 1964, we've existed as an independent university, and currently we have more than 27,000 students in total, of which about 2,500 are graduate students. In the two pictures that you see on the slide, on the one above, there is a picture of Johnston Hall, one of the nicest buildings on campus, and the one below is the picture of our mascot, the Griffin, which was actually built last year to celebrate the 50th anniversary of the University of Guelph. All right, so after this brief introduction about myself and my university, my employer, I'm going to talk a little bit about soybeans for the benefit of those of you who have not worked with soybeans or may not know much about the crop or the species of soybean, which has the name, the botanical name, Glycine max L. Merrill. And soybeans actually are a diploid crop. They are considered to be an ancient polyploid, and they have 2n number of chromosomes 40, and they belong in the family Fabaceae or Leguminosae, which means that it's a legume crop with the added benefit of fixing its own nitrogen from the soil, from the air. And soybeans were domesticated. The theories, current still valid theories, that they've been domesticated from the wild relative Glycine soja anywhere between 6,000 and 9,000 years ago. And it is currently the world's largest protein and oilseed crop because of its seed composition, which consists of about, on average, 20% oil and 40% protein. Soybeans were introduced to North America by a farmer who brought it on a boat, actually, to the United States, and he planted the first soybeans near Savannah, or in Savannah, in Georgia, in 1765. It took a long time for us to wake up and smell the coffee up here in Canada, so it wasn't for another 100 or so years that we actually brought soybeans from the United States to Canada, and I will tell you a bit more about that in a minute. Interestingly enough, soybeans were actually first brought to North America and then to Central America and South America, and Europe came later. So although Europe is geographically closer to China, it didn't see soybeans for much longer than the United States. So here's basically what makes soybeans so interesting to grow. It's mainly in the seed composition. As you could see here in the pie chart, there's about, depending on the source, it says about 40% or 42% protein, about 19 to 20% oil, about 35% are carbohydrates, and 3.5% are minor constituents. Of that oil, if you look at the other chart here, you can see that soybeans are considered one of the healthiest vegetable oils, because of a relatively small amount of saturated fatty acids, about 11% palmitic, 4% stearic on average, about 25% oleic acid, 52% linoleic, and 8% linolenic acid. Many products are made out of soybeans. It's considered that about 300, or actually more than 300, depending on the estimate, different products are made from soybean, and not all of them are food products. But initially I'm going to focus on the food composition, food qualities of soybeans. And soybeans are, besides being fairly high in terms of the composition of protein and oil, they're also rich in nutraceutical compounds with health benefits. Part of the health benefits comes from the protein itself, and there is an FDA claim that was issued I would say now about 15 to 20 years ago, which stated that a diet rich in soybean protein leads to a reduction in incidence of heart and cardiovascular diseases. There's other compounds that I will mention throughout my presentation that we've been working on from a genetic aspect and trying to breed soybeans that would have enhanced nutraceutical or functional food properties. The high-quality oil, as I mentioned earlier, because of its fatty acid composition, can also be altered genetically. When I say altered, I mean through mutagenesis and also through the plant transformation or GMO approach. Proteins contain all essential amino acids, so it's one of the best sources of protein there is. And in the pictures that I put here, you can see some of the many products that can be made from soybean, as well as soybean seed down here, and tofu as one of the soy food products. Okay, some of the other uses of soybeans. Soybean is used as a food, has been used for a very long time for traditional Asian products such as tempeh, miso, and tofu. And I'm sure those of you who have been to a Japanese restaurant have heard of miso soup, which is made from the fermented miso product that comes from soybean. Tends to be a pretty tasty soup. Even if you don't like tofu, chances are you might like miso soup. So there's also other products that are more geared towards the Western diet, such as soy drink or soy beverage. It comes with different flavors. Soy nut butter for those people who are allergic to peanut butter. Soy nuts, I put down my favorite, the red hot one, because of my Hungarian background. That's what I enjoy. I enjoy hot and spicy food, so that's an example of a soy nut that you might want to try. I would like to also say a few words about how soybeans actually made their way to Canada. It was due to the efforts of Professor of Field Husbandry, as it was called back then, in the Ontario Agricultural College by the name Charles Zavitz, and it would be probably called agronomy today. So he taught that course, or those courses, in our college at the turn of the 20th century. And in 1893, he actually went to Kansas, collected a whole bunch of soybean seeds that he saw being grown there, and brought them back to Canada and grew them in southwestern Ontario. Since Kansas is considerably more to the south than southern Ontario, only the very early segregants or early soybean plants were able to produce seeds, so he kept making selections without making any crosses, and through those selections he developed the very first soybean variety that was released in Canada for Canadian farmers to grow. It was called OAC 211, and it was released in the 1920s as a forage soybean. Soybeans on both sides of the border have been grown as a forage crop for a long time, basically until the Second World War, when the need for oil during the war and being domestically produced made soybeans into an oil crop. And they've been an oilseed crop ever since. In 2014, Canadian production area exceeded 2 million hectares, which is over 4 million acres. And most of the soybeans are grown in Ontario, about 65 to 70%, but we have seen a significant increase of soybeans being grown in Manitoba. And now farmers in Saskatchewan to the west of Manitoba are also considering growing soybeans as well. For those of you who are soybean breeders or have learned a little bit about soybeans, you would know that soybeans are actually grouped by maturity into 13 maturity groups, from 000 to group 10. And you might be surprised, some of you might be surprised to know that we can actually grow soybeans in a quite a range of maturities in Ontario, anywhere from 00 maturity group to the north, all the way to early maturity 3 groups in the extreme southwest. So just for orientation purposes, where I'm pointing with my pointer, that would be where Detroit is on the Michigan side. So in parts of Ontario, we can grow maturity group 3 soybeans. Most of the highest production of soybeans is in the maturity groups 2, 1, and 0. As you can see, that's the greatest area of southern Ontario. Again, for orientation purposes, this is where Toronto is on Lake Ontario. Guelph is noted here, and Rajcan, which is another campus of the university, is down here. I seem to be having some problems moving the slides. I have to click several times. I apologize for that. So moving on into how we do soybeans, or what has been done in terms of soybean breeding at the University of Guelph. The crosses, the really dynamic soybean breeding program didn't start until the late 60s and 70s. So most of the crosses were made from that point on. And they resulted in the release of over 115 soybean varieties that were developed at the University of Guelph. At that time, in the 70s, the only breeding programs were public breeding programs, and the private industry came online and started developing varieties much later. But the initial work was done by public breeders, some of whom were at the University of Guelph. And some of those varieties that have been developed, not all of them were equally popular or long-standing in the industry, but some of them have been. One example is OAC Bayfield. It's been around for over 20 years, and it not only was a very good variety, it's actually still grown, believe it or not, but it also contributed great genetics to other varieties because it's a high-yielding but also stable variety. That combination comes relatively infrequently in breeders' hands. So cultivars have been released every year since then, and some of the varieties that had OAC Bayfield as a parent have made a large impact as well. OAC Wallace and OAC Kent both had OAC Bayfield as a parent. OAC Drayton would be a grandson of OAC Bayfields, and some of the latest varieties of OAC Strive, OAC Eve were released this year. And OAC Kent and OAC Bayfield were both recognized as a seed with the Seed of the Year award, which is the highest recognition given to a publicly developed variety in Canada. So in 2 different years, these 2 varieties have been recognized. So right now I'm going to spend a little bit of time telling you a bit about how soybeans are crossed, because this is one of the hardest crops to make a cross with, and many people have tried to learn how to cross soybeans and failed miserably, and very few people actually can do that very well and efficiently. I'm very fortunate to have in my lab Mrs. Lin Liao, and she prepared these slides for me to show you how crosses are made in soybeans. Soybeans have beautiful flowers. They usually come in this kind of purple pink version or white. There's very few other combinations of colors in soybean, and as you can see, it's a typical flower morphology for a legume crop. And as you can see here, soybeans have 1 pistil with a stigma on top, 10 stamens, 1 calyx with 5 sepals, and 1 corolla with 5 petals. Those 5 petals are called standard, is 1, and there's 2 keels and 2 wings. And down below you see a soybean flower that's actually been prepared for crossing. So I'm going to walk you through these slides that Lynn kindly prepared and give you a sense of how we cross soybeans. Okay. So first of all, the first thing that a breeder has to do is to prepare the female flower. Obviously, because soybeans are a self-pollinated clays to gamete crop, if you waited until the flower is open, the flower would have been self-pollinated already, so we can't afford to do that. We actually have to go into a bud and emasculate the bud to prepare it for a cross. So choosing a flower bud is not easy. Choosing the right stage is not easy, and only an experienced person can choose the flower bud at the right stage to be ready for crossing. So the female buds that are actually ready for managing and emasculation tend to be swollen, and the corolla can be seen through the calyx. As you can see, corolla is here seen through the calyx, and that means that the flower is ready to be crossed, or the bud is ready to be crossed. In the next slide, I'm going to show you how the flower is prepared for crossing. First of all, the calyx is removed, then the corolla is removed. The 2 wing petals are removed with tweezers. And then the removal of anthers follows. The anthers obviously have to be removed in order to avoid self-pollination, and all 10 of them have to be removed to leave you with 1 stigma or pistil in the middle that will be ready to receive the pollen from the other parent. As Lynn pointed out here, it's very important not to injure that stigma, because if you injure it, there will be no seed as a result from the cross. And then in this slide, there's a preparation of the male flower. So you go to the other parent that you want to cross with the first parent, and you need to take the anthers that are already shedding pollen. So as opposed to the previous case, instead of going into the bud, you would go into the open flower where the pollen is already shedding, and then pick up the anthers. The stamens are picked up together with the tweezers, together with the pistil, and then they were brought to the female flower that was prepared earlier. And then the pollination occurs. You would basically just take that male flower anther structure and brush it against the stigma of the female flower to carry out the cross. And that finishes the crosses. So with that last step, the only thing left is to actually label the cross. And you can see here on the plants the crosses have been labeled, and usually write the female parent first, the male parent second, the date of the cross, and any other notes that you might want to put on that tag. So whoever comes to harvest the F1 seeds will know what the cross was. So it may look relatively simple if you look through the slides here, but it's an extremely difficult job, and very few people can do it successfully and efficiently. So I will just at the next stage I will walk you through the breeding program as we do it. It's certainly not the only way that soybean breeding can be done, but I wanted to illustrate it to you how it is done in my program. So typically we do a large number of biparental crosses where you cross just parent A and parent B with complementary traits to produce the F1, and then the F1 plants are grown to produce the F2 seeds, and we apply single seed descent method to F4 or F5, and we end up with releasing a variety at F9 or F10. So I will go into a bit more detail in the next few slides to illustrate that. So there seems to be a lag with the forward button. I'm sorry for that. So we make approximately 200 crosses per year, and when I say 200 crosses, I mean 200 cross combinations between different parents. It doesn't mean it's 200 attempts. It's probably 3 or 4 times more attempts to make a cross. And since our goal is to obtain about five F1 seeds per cross, because one pod would have 2 to 3 seeds, we usually cross, for one cross between 2 parents, we usually need about 3 pods, 2 to 3 pods to achieve the 5 F1 seed goal. So all crosses are made in the growth rooms, and the picture of the growth room is shown here with my former technician and a student who was helping her. So these are very good facilities that we have in the crop science building in our department, and they are used in the winter when we make all the crosses to do the crossing in soybeans, and other programs use them as well. So when the crosses are done in the winter, the F1 seed is usually harvested in April, and it's planted again in the growth rooms to produce the seedlings, and then the seedlings, when they reach the first or second leaf stage, we take them in these flats that you see down here. We take them to Rajcan campus, which is about 200 kilometers or approximately 220 miles southwest of Guelph. Why do we take them there? Because we have more heat there. The plants will mature faster there in the fall. And it's also an irrigated nursery where the F1 plants are space planted to produce as much F2 seed as possible. The F2 seeds are produced and harvested in October, and then we bring them back to Guelph, where they are packaged into 100 seed envelopes, about 500 seeds per cross, and then they are sent down to Costa Rica to our winter nursery for a 2-generation advance by single seed descent. So most of you who have taken plant breeding courses would be familiar with this schematic of single seed descent. I believe this was a schematic from Walt Fair's textbook on plant breeding, and it just shows the concept where One is actually not making any selections all the way until about F4. The main goal is just to maintain the population size per cross combination and to drive towards homozygosity as fast as possible. And when we're at F4, it's about 87.5% on average homozygosity. The selections we make at that stage are more meaningful to us because what you see at that stage is similar to what you get in the following year, as opposed to starting to make selections in a highly heterozygous population such as F2. So the rest of the schematic I'm going to explain on the example of our own breeding program. So this is what I call the soybean breeding path. It happens every year. In October, the first arrowhead is leading to Costa Rica. That's a blow-up of the map of Costa Rica, a Central American country. And then, so basically in the fall, we send our soybeans on a sabbatical to Costa Rica. They go through 2 generations, enjoy the tropical weather there while we are freezing up here in Canada, and then they are sent back in April back to Canada, and we plant them as F4 seeds the following year. So basically F4 seeds come back usually in late April, early May from Costa Rica, and then they're planted in our nursery at the Woodstock Research Station about 1 hour west of Guelph. And at that stage we actually see our progeny for the first time, and we start making single plant selections. The nurseries in Costa Rica are located in 2 different locations, and some of them are actually pretty close to beautiful beaches such as this one. But unfortunately, I haven't made a trip down to Costa Rica because we have a great cooperator who sends us the seed. I hope to go and see that beautiful country sometime and see what our soybeans look like. This is actually a picture of our own soybeans as they grow in Costa Rica. So why do we go there? Because we can get in as little as 6 months, we can get 2 generations of soybeans, and that saves us 2 years. If we had to grow the same soybeans in Canada, it would save us 2 summers. And we are not unique in this. Most of the breeding programs use winter nurseries to speed up their breeding process. So the F4 seed that comes back from Costa Rica is planted in the F4, or what we call the Costa Rican nursery at the Woodstock Research Station. There could be anywhere from 40 to 50,000 F4 plants in that nursery, depending on the production in Costa Rica. And from those plants, we select about 4,000 to 6,000 single F4 plants, basically just based on visual agronomic appearance. There is no yield measurement that's meaningful at the single plant stage, and then those plants are bundled, brought back, threshed through the winter as single plants, and each plant, the seed from each plant, individual plant that was selected, is going to be planted in what we call the F5 hedgerows, or single row plots about 4 meters long. And the selection at that stage, at the F5 stage, is based on maturity and plot appearance. So this is what happens at the F6 stage. So F4 derived F6 plants are grown in preliminary yield trials. It's a full-size 4-row plot that is grown at our research station, and the plants were grouped by maturity. So depending on the maturity of the plant, we grow them at one location, one research station or another. And basically we grow 500 to 700 plots at F6 stage, and we make selections from those plots based on yield. So the first time we make yield selections is at F6. We could have measured the yield at F5, but I simply don't trust the 4 meter long one single row to estimate yield. So in the F6 stage, preliminary yield trials, it's an unreplicated, completely randomized design trial with repeated checks. 5 to 6 checks are repeated about 3 times and scattered throughout the plot area, so we can measure the variation in the field. From those plots, about 700 plots, we select 150 to 200 based on yield to go into advanced yield trials in 2 locations. each with 3 reps each. So the picture here is an aerial picture of our plot area at the Woodstock Research Station that was taken by Google Earth, just to give you a sense of what the plots look like from the air. And this is what I call the home stretch. From those 120 to 150 plots, we can select about 30 to 50 lines to test them in multi-location trials across Ontario over 2 years, and sometimes we put them into trials in Quebec and Manitoba, as well as into the uniform test on the US side. So those lines then lead to the production or development of varieties, and breeder seed production usually is initiated after the first year of multi-location trials if the line looks pretty exciting, or after the second year if it looks better than the checks, but it's not maybe 110% higher than the checks in yield. So this means that on average we release 3 to 5 cultivars to the seed industry. We are not in the business of seed sales, so all of our varieties that are released are actually handed over on a bidding process to seed companies who then take care of distributing that variety. So this is why I think soybeans are exciting, an exciting crop to work with, mostly because of the seed traits. In my time as a breeder, I've been breeding mainly oilseed crops. If you looked at the first slide, there's a pattern there. Although I've switched crops over during my career, I've always stayed with the oilseed crops, and that means sunflower, canola, and soybeans. So I've always been excited about the opportunities to modify the seed traits to fit different needs in the industry. So some of the traits that we've been working on in soybean have been fatty acids, such as low linoleic fatty acid, low stearic fatty acid, high linoleic, isoflavones. I've had 3 students working on isoflavones, and I'll tell you in a minute what the isoflavones are important for. And then tocopherols. I've also had 3 different students working on tocopherols or vitamin E enhancement in soybean seeds. And the most recent interest has been in saponins, which are handled by a graduate student, Ed McDonnell, who was hired last year. And the reason why we're interested in saponins is because of their anti-cancerous and antimicrobial effects that they have. And soybean seeds have them. And we're just thinking about how do we change the profile of saponins to make them even healthier for the consumer. So the excitement here comes from the fact that especially the bottom 3 traits or mostly secondary metabolites can be enhanced in soybeans to improve the nutritional or nutraceutical value of soybeans. In other words, to improve the health benefits that come from consuming soybean seeds. And so basically what we are looking at is, instead of looking at soybeans as a commodity, which they are, and they are marketed on a regular basis through the Chicago Board of Trade that determines the price daily of soybeans, we are thinking of how can we as breeders add profit to soybeans by differentiating ourselves in the marketplace, which we see as an opportunity. And it's not easy to do, but it is interesting to do, and it's fun as well. And so basically we're shifting our paradigm from of looking at soybeans as a commodity to the development of niche markets and specialty value-added products that would have soybean seeds with added value through traits that have been enhanced to impart different attributes to the seeds. So this is illustrated by planting of some of our various breeding lines at the farm of Peter Hannum near Guelph. One year he asked me for seeds of various projects, from various projects that we're working on, so gave him the seeds and he planted them on his farm. And you can see here examples of high isoflavone soybeans, high vitamin E soybean, high oil soybeans, high sugar, especially for soy milk, that's important, and different fatty acid composition in soybean seeds. So in this range of different compounds, most of them being secondary metabolites in the seed, I'm just going to give you one example of soybean isoflavones that we've been working on for about 15 years. And why are they important? Mostly because they are recognized for their health benefits. They are naturally occurring in soybean seeds already, and they have been associated with the prevention or suppression of some types of cancer, especially colon and breast cancer, reduction of incidence of osteoporosis, heart disease, menopause symptoms. All of these things, all of these effects are imparted if a person consumes soybeans as part of their diet through their lifetime. So it's certainly not a medication. It's not a cure to any of these diseases. But if you have soybeans in the diet, the chances of you getting these diseases are actually reduced. So the compounds are fairly complex, and they are produced. Soybean isoflavones are produced from the phenylpropanoid pathway. And you see here a structural chemical formula of genistein, one of the three main aglycones of isoflavones: daidzein, genistein, and glycine are the main aglycones. And I actually removed the phenylpropanoid pathway slide because it's quite complex. But what I wanted to say is if one is working on a trait like this, because of the complexity of the pathway, you're basically forced, whether you like it or not, to work with multiple loci or multiple genes that are influencing the production of these compounds in the plant. So it's not easy, but molecular markers tend to help in that work. So another example I wanted to give is some of the modified fatty acid composition soybeans that we've been working on. You may or may not be aware that Ontario is one of the largest producers of automobiles in North America, and there's a number of different plants that manufacture cars, vehicles in Ontario, especially in southern Ontario. Some of them are listed here. Toyota has a factory plant, Ford, Honda, and General Motors all have factories in this stretch from about Detroit to about Oshawa up here in southwestern Ontario, in southern Ontario. And so the reason I mentioned that is that there's an opportunity here for soybeans and for soybean breeders, because when we develop soybeans with specific fatty acid profile, they are interesting to automotive industry because they can produce bio-based renewable. They have a renewable source of raw materials that can be used to produce polyurethane foam that's found in car cushions, and seat cushions, and head restraints and armrests of vehicles that are manufactured by the different manufacturers of automobiles. So we've actually done a bit of work in high linoleic soybeans, and I'm going to illustrate that in the next couple of slides. And two students have been on that project, Joel Hemingway and Sarah Parkinson. So this is taken from Sarah's master's thesis, where she looked at high linoleic soybeans compared to Czech varieties, standard soybean varieties that are grown by farmers, and what she found was that the highest yielding in her panel of varieties, the highest yielding variety was OAC Kent, but as you can see, RG25, one of the high linoleic acid soybeans, was much lower yielding. However, if one compares the composition of the seed, the highest amount of linoleic acid was in RG25. There were a couple of others that were higher, but they were very low yielding. So RG25 had the seed yield that was relatively close or comparable to OAC Kent. Well, actually closest to OAC Kent compared to other varieties. And because of the high linoleic acid content, one can produce more linoleic acid per unit area of production by growing this variety. However, we're still not happy about the yield of RG25, so we made crosses to other varieties, high yielding varieties, to get closer to the high yield that other varieties have with the same fatty acid composition to increase the production of linoleic acid for the auto industry. All right, so those varieties that we looked at, RG25 was the most promising variety, and it was tested for polyurethane production to be used in the auto industry. The environment had a significant influence on the total seed yield and seed linoleic acid content, but the genetic control was the main factor in determining high linoleic acid production. There was a strong negative correlation between linoleic acid content and seed yield in this material, and there are different causes, potential causes for that, because independent mutations may have been generated by ethyl methane sulfonate mutagenesis, which actually led to the development of this high linoleic acid trait. Linkage of mutant fatty acid alleles may have been a factor if they were linked to unfavorable yield alleles. And pleiotropic effects of mutant fatty acid alleles may have been a factor as well. So our goal was back then to incorporate these mutant fatty acid alleles into high-yielding soybean background. And we've made progress in that. So the more recent variety, OAC 1355CHL, which stands for high linoleic, had about the same amount of linoleic acid as RG25, but much higher yield. And when some pilot study analysis was done, as you can see, the arrow is pointing to the amount of double bonds per increase compared to commodity soybean. There's other soybeans that are produced by different manufacturers and different companies, high oleic acid soybean. All of those have lower amount of double bonds per unit of oil. And our high linoleic soybean had 12% higher than the average soybean, commodity soybean oil. So that's of interest to the automotive industry as well as the coating industries. So in the last part of my presentation, I'm going to talk a little bit about a couple of projects, just briefly, of some of my students. There's many of them, but I had to pick a couple, so I've picked master student Torrin Boyle. So you can see him here in the picture with his brother selling organic produce. He comes from an organic farm near Hamilton in Ontario, and he joined the lab a couple of years ago, so he's working on his master's degree. And so just as a background to say that organic agriculture is a management-intensive approach to farming which seeks to take advantage of agroecosystems to enhance crop production. And it's characterized by complex rotations, biological pest control, and cyclical fertility systems that evolved that are also involving integrated livestock systems. So how did it come about that I became interested in organic soybean production? It's in part because of Torrin himself. So the student came and he was interested in pursuing a master's degree, and I gave him a different project. He asked me if he could do something to do with organic agriculture, so we basically designed a project for him. And so why would anyone want to breed soybeans for organic production? Organic farmers are currently utilizing conventionally bred varieties, which may or may not be a good thing. It means those varieties have never been exposed to organic production systems and may not represent the best genetics that can be grown by an organic farmer. There's money in organic agriculture. The organic soybeans are sold sometimes 2 to 3 times higher at a 2 to 3 times higher price. And so why shouldn't those growers have the best genetics just like the rest of the soybean growers do? And so crops in organic systems encounter more environmental stresses, which means that there's possible different environmental influences. Increased weed pressure is a problem. Reduced nutrient availability is a problem because one can't use inorganic fertilizer. So the question was, is the genetics different for optimal organic performance in the current varieties, or is it the same? So in that project, we've kind of split it into 2 experiments. One involves 30 Ontario soybean cultivars, diverse cultivars that were planted in an RCBD design on an organic farm and a conventional farm. And standard and organic traits were measured throughout the development of the growing season. And the second is a breeding trial where we picked 2 crosses that were made in the program between food-grade soybean varieties of diverse pedigree and both were selected under each environment, and selections are compared. And I'll show you some of the preliminary results. Just to mention some of the organic farming traits that one has to measure, which they may not have to measure in a conventional system. Weed suppressive ability is measured early in vegetative growth. Nutrient use efficiency is measured usually at R5, that's a reproductive stage 5. Root morphology was also measured in a Torrance test in the top 20% of the soil at R5. So the locations were actually fairly close as we could find, because we already have our conventional location at the Elora Research Station, which is shown here. And that's Elora, Ontario. It's about half an hour drive northwest of, mostly north of Guelph. 25 minutes apart is the Moorfield location, where there is Mapleton Organic Farm, and that's where the organic trials were grown. So among other traits, I don't have enough time to talk about everything, obviously, but I'll just do a little bit of, mention a little bit of data that's been produced in the first year of trials. In Elora, at the conventional site, for all root morphology traits, there was no difference among the cultivars, which kind of surprised us a little bit. At the same time, however, at Moorfield, the organic site, there were significant differences between the varieties for all the root traits. So the fact that we grew these same varieties in an organic site meant that the roots were developing in a different way. And also what we did was we did a ranking and rank correlation between the in terms of the performance between the two locations. So as you can see, Moorfield was the organic site, Elora was the conventional site. There's a lot of crossover effects. The correlation is significant at 0.55. However, imagine if you were a farmer growing a variety DH530. If you grew it at the organic site, you would have good luck because it's one of the highest yielding varieties in the organic farm. But if you grew it at the Elora station, it's pretty low yielding. So depending on whether you grow the same variety in one production system or another, the performance could vary. At the same time, if you grow something like OAC Sunny variety, the performance is pretty good in both locations. So in terms of the breeding populations, that's the second experiment. 2 crosses were selected. About 150 single plants were harvested from each cross. OAC Calypso by DH618 and OAC Sunny by S08T6. So at the F5 stage, we grew them in single rows, and selections were based primarily in terms of visual performance by Torin and myself, and we applied about 25% selection pressure. And what did we get? Interestingly enough, out of 38 varieties that we selected from the first cross, only 21% of the varieties selected at each location overlapped. So, which means about 80% of the varieties that we liked were different between the two locations. In the second cross, it was even more pronounced. The selection pressure was 26%. We selected 14 rows. 42 rows, and only 14 or 33% of the lines were the same between the organic site and conventional site. So it tells you that if you are selecting visually at one or the other site, you might be missing out on a lot of lines because the overlap is not that great. And the trials are continuing now into the yield trial stage in this year. The second project I will tell you a little bit about is by Robert Bruce, a PhD candidate in my lab. He's working on understanding the effects of breeder selection on elite soybean germplasm diversity. And so basically, as it stands currently, most of the breeding programs use elite germplasm that's recycled for crosses year after year. Most of us cross elite by elite germplasm. We rarely cross with the exotic germplasm because it may slow down or or reduce the progress from selection in cultivar development. And parental selections are made purely based on agronomic performance. Very few breeders actually look at the pedigree information to select parents for crossing. So basically, we're interested in linkage blocks in different chromosomes of soybeans. If you look at this slide, you will see there is a section among these 5 lines that's illustrated with low diversity at this block or linkage block, and there's a section with high diversity where the allelic content is quite different among the different lines. So a couple of questions can be asked. Is there no favorable block in these locations? That's why there's high diversity. And on the other hand, is this the best block already that's been fixed, and there is no further progress that can be made if we change the allelic composition in this block? So those are some of the questions we're trying to ask in Robert's PhD project. So the questions are, have breeders' selections reduced diversity at loci for important agronomic traits in Ontario soybean germplasm? Is allelic structure conserved between breeding lines and cultivars for the same traits? Can targeted introgression of diverse lines from the plant introductions further increase yield without compromising or diluting the selection signatures that have already been developed over decades of breeding? And for that, Robert has a unique germplasm made of a panel of 192 lines representing about 60 or 70 years of breeding at the University of Guelph, about 96 lines from Rajcan campus. That's a full season soybean breeding program of maturity group 2 and 3 that's led by Dr. Eskandari. And ours is basically a short season to medium season breeding program. We have quite a diversity of lines, cultivars that have been released and grown, breeding lines, Chinese germplasm, ancestral lines, tofu, natto lines, and even some interspecific recombinant lines are part of these panels. So one of the questions we are asking is, has selection for a trait in both programs altered the diversity in the same pattern or different pattern? So for that, he's using genotyping by sequencing approach. We send our samples, the DNA is extracted here and sent, the samples are sent to Genome Quebec lab in Quebec City, and they're run through the Illumina, actually HiSeq, which is their latest machine, and then the bioinformatics is applied to filter the data and give us SNP calls that we use in the analysis. So from the wealth of information that he's produced, I'm only able to show you a tiny bit. I'm talking about Robert's project. So one of the things we're trying to determine is identify linkage blocks and their pedigree origin across the breeding programs, find out if there are any signatures of selection, and I can tell you already there are. What genomic regions have altered diversity compared to other regions? Can we associate diversity in genomic regions to traits of interest? So this is a circular tree diagram that is part of the data that Robert has generated. It just shows the first set of 96 lines that were clustered together, and there's definitely a population structure involved and quite a lot of diversity in this panel because of the way that we put it together. So most of the lines are actually consistent in terms of their pedigree information and molecular marker diversity that came from this analysis. So the project continues on, and in another talk at another time I might be able to give you more information about it, but because of the limited time I will have to stop talking about it right now. So in the end, I just want to acknowledge, if I wanted to put a list of all the people that have been contributing to my lab and in various projects, and both the current and past grad students, it would make for a very busy slide, so I'm just going to thank all of them together for their contributions, and there have been a number of grad students, technicians, postdocs, and collaborators nationally and internationally that have contributed, as well as solid funding that I'm fortunate to receive from seed companies, from the Ontario Ministry of Agriculture and National Sciences and Engineering Research Council of Canada. And with that, I'm going to end my talk, and thank you all for your attention and for your participation in this webinar series, and especially for taking part in my own webinar. So with that, I will pass it on back to Anna. Thank you.
Thank you, Dr. Istvan. Right now we have a few questions. Just as a reminder, if you have questions, please type them in the question box. So the first question we have is, could you describe how breeding activities are supported at the University of Guelph?
Certainly, I'll be happy to. We often have these debates in the public sector as to, sometimes we receive criticism from private industry as to why are we developing varieties when there is private industry that's doing the same thing. Well, first of all, we don't necessarily work on the same traits and on the same germplasm. Most of my breeding program, I would say about 80% of it is devoted to conventional soybean variety development, and most of the companies are actually working on developing glyphosate or Roundup tolerant varieties. So there's not as much overlap between what market we are addressing and what market the private companies are addressing. And those companies who are selling conventional seeds for food grade production for export markets to places such as Japan and other Asian countries are interested in us continuing the work on the variety development in the conventional arena. And once we develop varieties, we release them to those companies. And because of all that, going back to the original question, I will say that we have enjoyed great support from mostly from the companies that are actually picking up our varieties after they've been developed, and they are marketing them worldwide. They're also involved in testing them as far as Russia and Ukraine, and also down south in some of the U.S. states. And it would be, just to add one more thing, I'm also fortunate to have good support from the Ontario Ministry of Agriculture and Food and Rural Affairs to my research program, and they are also paying the salary, part of the salary for my position, and also providing research station support.
Okay. Thank you. The next question is, what are the greatest contributions to breeding research that have come from the University of Guelph soybean breeding activities?
All right. That's a fair question. So I would say the greatest contribution is the development of germplasm for short season soybean areas. So basically, I didn't have enough time, but there was a project that we had in my lab, and Robert is actually continuing following up on that project, where we looked at the contribution of ancestral lines and ancient varieties to northern regions of the U.S. and Canada. And there's one variety that was actually brought from Sweden. It was called Fiska B5, and large linkage blocks from that Fiska B5, very early short season variety that was brought from Sweden was found in many different varieties that were released in Ontario. And so the variety was actually picked up by my predecessor, soybean breeder at Guelph, and some of that earliness that allowed us to move the soybeans north and west within Canada have come as a result of breeders' efforts before me. And beside that, I would say the other contribution is we are still developing varieties that are popular among the farmers. It's estimated that of the 3 million or so acres of soybeans in Ontario, about 25 to 30% are conventional soybeans. The majority of that acreage is actually planted to soybean varieties developed by Guelph. And beside that, some of the companies that are using low linoleic alleles in their breeding programs to develop high stability non-trans fat soybean oils have been using alleles that were a result of mutagenesis work done by my predecessors at the University of Guelph as well.
Thank you. The next question is, as you shift to a value-added focus in your breeding program, do you think it will change how you commercialize material? Specifically, do you feel like you will be able to continue with the model of licensing or selling cultivars to seed companies, considering that they may potentially have less interest in the pursuing niche markets?
It's an excellent question. I'm glad that the person has asked that question. And if you think I don't think about that on a daily basis, you're wrong. I do. Part of the issue that a breeder has is most of us work on a wide range of different traits, but what does it matter if nobody wants to pick up those traits? So I constantly think about that, and it's important to stay in touch with everybody and do your networking to find out who might be interested in the traits that you are working on. In that, I received great help from what we call our Catalyst Center, which is actually involved. It used to be called Business Development Center at the University of Guelph. They're involved in making connections between researchers in terms of their inventions and the potential users of those inventions. So they provide great support to us, as well as an organization called Soy 2020, which is an organization that also is involved in connecting the researchers with potential users of their technology. So what would that mean in terms of change as far as how do I access the markets if the companies are not interested in those traits? Well, we may not work with the same companies. Sometimes in the case of traits that are important for automotive industry, it might be automotive parts companies that would actually be interested in licensing those varieties and working with the seed growers or seed companies to produce varieties that would be good for polyol or polyurethane production. So it has to be handled on a case-by-case basis. In terms of nutraceutical traits with health benefits, the healthy food markets and food industry may be more interested in that, and they generally would connect with the growers who would be growing those varieties that we developed and paying them a certain premium for growing varieties with added traits.
Thank you. The next question is asking if the stink bugs are a problem in soybean crops in Canada, and if there's compounds associated with isoflavonoids that are related to resistance to the soybean insects. It would seem to be an advantage to have those compounds. In addition, we can use these genotypes. Can we use these genotypes to be included in the organic systems production? And have you been working this line of research?
Okay, that's quite a loaded question. I'm not sure if I will be able to memorize everything that's been asked, but I will start from the sting bugs. First of all, so far, luckily, in one way, it's a good thing that we have different pests and diseases than the southern U.S. because we don't have to deal with the same issues. And a stink bug would probably be one of the insects that's not had a great impact on soybean production in Ontario so far. Their population is on the rise, but I don't think it's at economic levels right now to justify spraying with insecticides or anything of that sort, or to justify setting up a separate breeding program to deal with stink bug resistance. So we do have a bunch of other diseases and pests that are more important to our production region, including soybean cyst nematode. That's on the rise, and it's been moving north and west, north and east throughout Ontario, county by county. So every one of us has to work on SCN resistance, and there is also white mold, Phytophthora, root rot, and a number of other diseases that are more of a problem than sting bugs. Are isoflavones potentially useful for dealing with sting bugs? I would say there is a potential. The one good thing about isoflavones is that they are produced through the phenylpropanoid pathway, which leads to the production after isoflavone step. The next one of the next compounds would be phytolexins, and such as glyceolin, and those phytolexins can actually have anti-insecticidal effects or anti-fungal effects. So I'm not doing that research myself, but I have a student that I'm co-advising with Dr. Eskandari. Together, his name is Adam Carter, and he's going to look at segregation for isoflavones in a soybean population, where we will check whether the different levels of isoflavones are contributing to enhanced resistance to soybean cyst nematodes. So not necessarily stink bugs, but other pests and diseases may be related to isoflavones in soybean.
Okay, thank you. A few more questions. How can we increase genetic diversity since it is a very tight bottleneck selection that has happened in the past?
It's a great question. That's one of the things that, or tasks that I've given to myself when I started at Guelph. I was worried about some of the reports that came out in the late 90s from Tommy Carter's lab and Clay Sneller's and some other labs that indicated the diversity of soybeans was very narrow. There's about 35 ancestral lines that make up about 90% of the genetic diversity of North American soybeans. So when one thinks about the origin of soybeans being in China, the fact that we are relying so much on 35 ancestral lines when there's tens of thousands of different genotypes growing in China is a concern for us. And so what I'm trying to do myself about that is I've been collaborating with Chinese scientists from northeast provinces in China, Jilin and Heilongjiang. And those would be the provinces where they grow soybeans of similar maturity to Ontario, so we can exchange germplasm. And we have mapped some QTLs that are universal, expressed in both mega environments, China and Canada, or they are specific to one mega environment or the other. So how can we increase it? We basically have made some crosses between elite lines from China and elite lines from Canada, and even without any back crossing to the Canadian line, we've released some varieties that are highly diverse compared to the rest of the germplasm, and they have contribution from alleles from China, and they are also high yielding as well. So we're making progress in increasing the diversity, and that's also been noted by the private industry, and some of them have contacted us to access that germplasm to increase diversity in their programs as well.
Okay. The next question is, how is exactly soy oil used in automobile? Is it used to run an engine or any other way? Could you explain that a little bit?
Yeah, for sure. So first of all, soybean oil can be used directly with some processing as a fuel, as a biofuel. Biodiesel, I apologize. So biodiesel can be produced from soybean oil, so that would be more or less a direct use of soybean as an energy source. What I was referring to when I talked about the high linoleic acid project is using soybean oil that has a modified fatty acid composition, which is more suitable for further processing into polyurethane. So, for example, polyols are produced from oils that have multiple double bonds, and the optimal number of double bonds without making soybean oil unstable is 2, and linoleic acid happens to have 2 double bonds. And so what we have done is we have increased the linoleic acid from 52% to about 70%, which makes it a better raw feedstock for the polyol and polyurethane industry. So when they take that oil, process it into polyols first, and polymerize it to polyurethane, they can produce foams that become part of interior car parts. So because this is a constant source, soybeans can be grown every year. Instead of using crude oil as a feedstock, we can use soybean oil and have a renewable source of polyols and polyurethane year after year.
Okay, and the last question. We know that oil and protein have a negative relationship. What is the best strategy to increase both at the same time?
That's a great question. I'm not sure if it's actually possible to increase them at the same time, although I'm aware of some studies where if one looks at the correlation, it's usually very strong. and strongly negative between protein and oil. So by increasing the protein content, the oil content tends to drop, but it's not linear. And if one is to plot a population that's segregating for oil and protein, there are certain dots on that plot where you would find rare segregants that are increased in both oil and protein. It's difficult to do, but it's not impossible. So the only explanation that I can offer for that is keep in mind that soybean oil and protein together make up about 60% of the seed. About 35% are various carbohydrates, raffinose, stachyose, and other carbohydrates. If part of the volume of the seed that adds to protein and oil comes from that 35% of carbohydrates, that would be one way of increasing both the oil and protein. But there's probably a certain breeding or genetic or physiological limit to how far we can reach with that.
Okay. All right, everyone. Unfortunately, we're out of time, but I would like to thank you all for your questions and for attending the webinar. As a reminder, recording this webinar as well as all the other webinars from the series will be available at eextension.org/plantbreeding genomics. I believe the URL is also on your screen. And thank you very much for joining us again, Dr. Rajcan, and thank you very much for our audience for coming.